Alrighty, who's ready for a treat? Okay, so thank you for coming back and in this abbreviated lunch period. We are really thrilled to have 4 student scholars with us today who answered the call of a call for participation, which we gave them just a very short notice and said, who's out there who has a project who would like to speak on their research at the GTI Symposium? And 4 people immediately said, whoa, I've got something. So we are thrilled to be able to share them with you. They'll each have about 10 minutes to present, and then we'll have a Q&A afterwards. But we have 4 students, 2 undergrads, 2 grads, so nice variety, and also a variety of topics. So again, from across the disciplines, demonstrating once again the importance of GTI for the study of culture and science of tea. So our first speaker is Mr. Quanrun He, and his topic is, where is the future of Chinese tea drink? Quanrun, please come on up.
All right, we can see on the picture on the PowerPoint. I think you guys have already seen this picture. So the one on the left is actually a now rapidly growing Chinese tea brand, which is called He Tea. So that is the one that people usually talk about a lot in China. And on the right is the one like we're really familiar with, which is Starbucks. But we can say like people just lining up in front of He Tea, but not the Starbucks. So if that means like tea is getting back its popularities or is competing over Starbucks? Because as we know, like Chinese Starbucks really have a large market shares like in past several years. So that is really an interesting question. So in order to answer that question, we're gonna first examine some data. So actually on this paragraph, we can say the sales revenue of Starbucks is increasing rapidly like during past several years. But however, we can say the growing rate is actually decreased drastically since 2013. And that year is particularly interesting because this is exactly the new tea drink coming to the market and competing with Starbucks. And then we can say it's actually taking over a large share of market of Starbucks. And then we can say from here it's 72.9. However, under 2016, it's already come to 19, which means even though Starbucks is still really competitive in Chinese market, but it was challenged by the new Chinese tea drink. Let's say this paragraph, which makes things more interesting. Because as we can see, these 2 on the left, this is the growth rate of the coffee as a whole. And the red bar, the blue bar actually represents the chain store, the individual coffee store, which is a negative growth. But even though the big brand like Starbucks, like big coffee drinks, they tend to lower their growth rate. But tea is totally a different story on the right, because we can say like no matter you are a small tea house or like you are a chain brand like like He Tea or something, you're all making money and you're really growing rapidly during past several years. So that means tea is actually come to the market and play a more important role in in Chinese beverage market. Okay, so we may ask, even though like tea was so successful, like it's got like massive commercial success, why people still criticize them, or why we still think it's not gonna like really lead us to the right place? So there are 3 main criticisms of Chinese new drink. So the first one is actually the most central one. That is when their brand selling their teas, they actually place their social characteristic over their product itself. Because when people lining up like just for the tea for hours, and then they're gonna post their picture to their social mediums. So it's actually tea itself was actually less important than its social characteristic. And then the second one is the overly commercialization and the advertisement, because as this tea brand growing, the strategy they usually use is hiring people to lining up in their in their stores, so they can actually gain a lot of attractions. In this way, they can sell their product and getting its popularities. But this strategy has been criticized by so many people for so many times. So I think this is really the problem of their strategy. And then the last one is actually not that important, but it still really influences our customer experience. Because usually when you want to get a tea, you want it to be fast, maybe like within 5 minutes. However, when you're trying to get a tea from Haiti, it usually takes you more than half hours or even like 1 hour. So it's quite incredible for many people who just want a convenient or like decent tea. So these three factors actually make Haiti the center of the criticism. Here, I want to propose a really interesting possibility of future of Chinese tea. And I'm going to introduce a small tea house, which is in Sichuan, Chengdu. That tea house has been opened in more than 150 years. So when it was open, it was still the Qing dynasty of China, which is quite a long time ago. And different from another tea house, which in our stereotype of tea, we usually think tea is kind of quiet drink. You just sit there and have some ceremony and just like that. But this tea house is totally different from our normal impressions, because this tea house actually reflects intense, vibrant culture, local tea culture. Because people just take their tea as their partly of their everyday routines. So when they drink teas, they actually regard it as like just American people drink coffee. So in the background, the picture is the whole view of the Guanyinge tea house in China, and then you can see the wooden chairs. You can see the like really colorful kettles. So these are all the part of their tea house display. So I don't know if I can play these videos, but I'm gonna try to play it because it's really interesting. As we can say, this is just give us an overview of what the tea house look like. Okay, since we cannot play it, so that's still fun. So this tea house, as I introduced to you, is really different from our traditional understanding of Chinese tea house. And then, so it's actually represents a future trend of their tea product. So what is really important is to think about how to put this small tea house setting in a commercial scale and actually make it to the commercialized scale, just like Starbucks make coffee. Because before Starbucks actually make a lot of standardization of coffees, people's understanding to coffee is actually really varied. They don't have really a definition of what is good coffee. They just have coffee in cans. So if we are able to put this tea house in a large scale and actually make it a representation of Chinese tea, I think that will be a really bright future of Chinese tea. So this is my talk. Thank you, everybody.
So, you know, Christmas every day, GTI, I keep saying this, I keep meaning it. So Quanrun is a food science major, and he just happened to wander into my office during office hours a couple weeks ago, and maybe it was last quarter, and sort of started chatting up and saying, you know, you were a tea, so I like tea. So we started chatting, and then he'd come back again for office hours and just sort of chat, and I'm going, you know, usually students want to know about their papers or whatever. And he was just like, you know, I'm just fascinated that you're doing tea. So when we had this opportunity to have student talks, he was like, oh, please, 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 and in the nicest possible way. But we are grateful. And another student who wandered into my office pretty much in the same way is our next speaker, Lydia Chin Yi Kao, who is a double major in International Agricultural Development and Communications with a minor in German. And so she wanders into my office and we're chatting about tea, and she's like, well, you know, Dr. Burnett, you know, how come there's no tea on the student farm? Why isn't anybody growing it? Well, because nobody's asked. And so she's asked, and so that is the basis of her talk today, growing tea plants in UC Davis Student Farm. Please welcome her.
So, hi, my name is Lydia Chin Yi Kao. Today, I'd like to talk about growing tea plants here at UC Davis, specifically in the ecological garden of the student farm. I proposed this tea project in fall 2017. Before I begin, I'd like to talk a bit about myself and why I became so fascinated and passionate about tea. I am a current sophomore here at UC Davis pursuing a double major in International Agricultural Development and Communications alongside with a minor in German. I think one of the most frequent questions I get asked is, what do you want to do in life? I will tell them that I'm particularly interested in getting into the tea industry. And the following question is, why? Well, here's a diagram that basically summarizes why. Long story short, tea covers many of my interests. For instance, agriculture, justice for marginalized and indigenous people, bridging of cultures, so on and so forth. As a Taiwanese immigrant who is half indigenous, I see myself as someone who can bridge gaps between the East and the West and the marginalized and the majority. We are in the age of globalization, and I believe that interconnectedness between people and cultures is important and key in the world we are in today. Before I begin the main part of my presentation, I'd like to acknowledge a few people who have been such great advisors throughout this process. First off, I want to thank Dr. Gaskell, who spoke yesterday about growing tea in California. He has been working with me via email with my hectic college schedule by providing me with so many resources from books to articles. Next is my ecological garden mentor, Julia Schreiber, who is one of the leading gardeners in the student farm. She is the one who has patiently taught most, if not all, my gardening skills and knowledge, and I am so grateful that I'm able to work with someone so experienced. Lastly, I want to extend my thanks to Professor Burnett. She provided me with so many connections, one of them being Dr. Gaskell, and most importantly, with our ambition and push for establishing a tea institute here at UC Davis, I will have not found my passion. The goals of this project for me is to process the harvested leaves into a consumable beverage. I specifically want to hold small tea workshops through the student farm. I also hope that I could spread more awareness about agriculture through this project. There are many goals that I'm aiming for, but may be altered as the project progresses, fingers crossed. On to the main part of my presentation. The logistics of establishing tea plants here at UC Davis. The two main conditions that differ from other tea projects are working with the UC Davis environment and abiding to the student farm principles. The main principles are to practice organic production, which means no chemical pesticides, fertilizers, etc., and how to be sustainable environmentally, which means it should be as productive in 50 years as it is now. This is the most basic outline I created that encompasses the timeline of tea cultivation. It has three portions: establishment of soil, irrigation, and climate, maintenance, and harvest. For today's presentation, I'll be discussing on the current plans of establishment, which will be implemented starting spring 2018, so next quarter, unless unforeseen events occur. Again, crossing fingers. Soil. We know that tea plants prefer acidic soils that are well-drained. Davis soil is around 6.8 pH and is silty loam, of course with some variance, and is generally well-drained. To lower the soil acidity and maintain it, I will add either compost, kelp, fish, and/or sulfuric fertilizer from the student farm. I will be adding sphagnum peat for quick acidification around the roots when transplanting them into the ground. Irrigation. Tea plants grow well when it has lots of water. It has been proven that drip irrigation been an effective watering method. And perfectly, it seems that closed spaced outlet drops is the best way to irrigate Davis soil. Luckily, the student farm does utilize drip irrigation. The drip irrigation is turned on manually every 2 weeks during winter, every 3 days or so during spring and fall, and every other day during summer. It stays on for approximately 4 hours every time for a deep, slow soak. I recognize that tea plants would like to be well watered, therefore I will adjust the time lengths as needed. Climate. For my research, tea plants can grow well in full sun, but partly shading them help their appearance. They cannot handle real winter. Davis has hot, dry summers and rainy, cold winters. This year, there were many foggy days in contrast to last year. The temperatures are somewhat extreme as well. From my personal experience, it's very hot here during summers, so to prevent potential burns, I plan to use student farm row cover called Tough Bell to shade them if needed. So what has been accomplished so far? It's been decided to bring in 3 plants as a start. Mature plants are preferred because I am graduating in 2 years. I would like to at least have 1 cup of processed leaves before I leave. Dr. Gaskell will also be providing some of his seeds, so there could be some experimental tea cultivation with the usage of the greenhouse. In addition, he, Julia, and I were able to look at the location together this past Wednesday where the tea plants will be established, which will be at the extended flower path of the ecological garden in the student farm. For the past few weeks, the intern and I have been weeding out an invasive plant, Lipa, and are still getting the site ready for the plants. Recognizing that this is only the start of the project, there's a lot to work on in the future. First off, I need to decide where I'm buying the plants from. Second, I need to ensure the plants are thriving and growing healthily. After a successful establishment, I'm hoping to bring in at least 5 more plants. Moreover, I need to read more about maintaining the plants, such as pest and disease prevention, pruning, so on and so forth. To recap, overall, I'm excited for this to be a learning experience for me. To repeat, I foresee trials and errors, and I will need to be extremely attentive to what the plants show if they're deficient in a micronutrient, burning, etc. I personally want to understand on a first-hand basis what tea industry workers experience in cultivating tea, and most importantly, I really, really hope to spread awareness about agriculture, which includes tea and the UC Davis student farm. I feel that in my generation, agriculture is truly underappreciated. I feel that people do not know how many hours are put into cultivating a successful crop and such a product, such as tea. I feel that there needs to be more recognition of the difficulty and hard work of being in such a field, especially for smallhold farmers. It's even harder when trying to grow organically. The list goes on, and I feel that Mr. Melican did cover very thoroughly the challenges of being in such a field yesterday during his talk. I'm hoping to spread more awareness to the public through providing tea workshops on campus while I'm still a student here at UC Davis and getting involved in the tea industry in the future. And again, I recognize that there's so much more learning that needs to be done. This is only the beginning. If you have any question, guidance, and / advice, here's my contact information. I hope that sharing with you all my background, my tea project here at UC Davis, and my hopes have made you all excited for not only this project, but also what Global Tea Initiative is aiming for. That is all for my presentation today. Thank you all for listening.
See why we love our students? So let's give a big hand of applause for our undergraduates because they both did really stellar jobs. All righty, so the next one is it Crystal or is it? No, wait. All right, so now we have our graduate students. Each of these students is working with one of our GTI faculty members. The first up is Ran Wei from China. She's a visiting scholar here for the year, working with Robert Hackman in the nutrition department. Her project title is Exploring the Anti-Cancer Effect of EGCG, a major component in T in breast cancer. Please welcome her.
Thank you, everyone, and it's my great pleasure to come here and to share my presentations with all of you. And these two people who have presentations before me is very interesting. And today, I would share with some data from the lab with you. Hope you will like it. Exploring the anti-cancer effect of EGCG, a major component in T in breast cancer. The breast cancer is still a worldwide problem. In US, the situations are more serious in Wisconsin, Missouri, Mississippi, Louisiana, and New Mexico. And among the races in US, we could see that the non-Hispanic white and black have the higher ratios. Have the higher ratios of both the incidence and the mortality than any other ethnicities. And in this study, we use the tea extract EGCG. It's a component extracted from tea and mainly from the green tea. And since it has the largest numbers of the hydroxy, it shows excellent effect in antioxidant by donating its own hydrogen atom to the free radicals. And since it is extracted from tea, so it's much more safe and natural. And as for the anti-cancer effect, however, there are already many papers published about EGCG and its anti-cancer effect, such as in the breast cancer model, in ovarian cancer, esophageal cancer, lung cancer, prostate cancer, hepatic cancer, oral cancer, colorectal cancer, and bladder cancer. Also, some scholars have verified that EGCG could also enhance the sensitivity of the cells to the chemotherapeutics like the cisplatin and 5-fluorouracil. Since the mechanism of why EGCG have the anti-cancer effect hasn't been clearly as yet, in this study, we focused on EGCG and its effect. Here is the structure of EGCG on the cell proliferation, cell apoptosis, and finally, we have examined its anti-cancer effect in vivo. So in the first part about EGCG and its effect on proliferation, here we have shown the cell viability. After treated with EGCG, we could see that the cell viability or the proliferation rate decreased in a time and dose-dependent manner. And you could see from the left pictures, the cell has lost its normal structures, especially in the higher concentrations like 160 and 240 micromolar group. So at that time, we would like to know why EGCG have the anti-proliferation effect in the breast cancer cells. Actually, in 1927, Dr. Warburg proposed that the mechanism of glucose metabolism is quite different in the cancer cells compared with the normal cells. As we all know that in the normal cells, the cells will primarily rely on the TCA cycle to get the energy, like the ATP. Only a small part of the glucose will turn into the lactate, and we call this process anaerobic glycolysis. But in the cancer cells, they instead primarily rely on the glycolysis, even there are enough oxygen exist. It's just the prospect of Dr. Warburg, and since the proliferation process is quite complex and So, we just examined the EGCG's effect from Dr. Warburg's perspective. And after the glucose has entered into the cytoplasm, it will turn into lactic with the help of many enzymes, especially the folate-limiting enzyme, the HK, PFK, PK, and LDH. And thus, the glucose and the lactic acid are the substrate and the product, respectively. We have measured the level of glucose and lactic acid in the cell medium after treated with EGCG. We could see that they show the opposite trend. It's because that if you added higher concentrations of EGCG into the cell, they will grow more slowly, and thus they will consume the less glucose from the medium, and much glucose will also still in the medium, and they will produce the less lactic acid. And we also measured the ATP production level in the cell, and it decreased after treated with EGCG. And then we measured the activity of the folate-limiting enzymes. It seemed that after treated with EGCG, the activity of HK, PFK, and LDH decreased. However, EGCG showed less effect on the PK. So in the first part, we concluded that EGCG showed anti-proliferation effect by modulating the glycolysis, or be more accurate by inhibiting the rate of the glycolysis. And in the second part, about EGCG and its effect on apoptosis. Here we have shown the process of apoptosis. The normal cell will lost its structure, and the cell will condense. After treated with EGCG for 24 hours and 48 hours, we could see that both the early apoptosis rate and the late apoptosis rate decreased. And in order to explore the mechanism of why EGCG have the pro-apoptosis effect, here we show the heat map of the apoptosis-related genes expression after treated with EGCG. The blue color reflected the decrease of the gene expression level, and the red color worse-wise. The expression level of survivin, BACO2, BACO-XL, and CMYC decreased. Actually, there are the anti-apoptosis genes, and other genes, most of them are the pro-apoptosis genes. So their expression have changed after treated with EGCG. And then the mitochondrial membrane potential, MMP, is also a key indicator in the process of apoptosis. And we have do the experiment by using the JC1 staining dye. Normally, the JC1 will aggregate in the matrix of the mitochondria and emits red fluorescence. And when the mitochondrial depolarization occurs, the membrane of the mitochondria will become more permeable, and the JC1 aggregate will disperse into the cytoplasm, and the red fluorescence will turn into the green fluorescence. We could see that after treated with EGCG, the red fluorescence disappeared gradually, and the green fluorescence become more obvious. So EGCG showed the pro-apoptosis genes effect by modulating the gene expression and the mitochondrial membrane potential. The third part, in vivo. Thank you for the mice. And after the mice have adapted to the environment for about 2 weeks, they were injected subcutaneously with 41 breast cancer cells. And 2 weeks later, the tumor appeared. The mice were injected intraperitoneally with drug, EGCG. And at the same time, we have set a group and injected them with the chemotherapeutic, the 5-fluorouracil, to be the positive control group. And after another 2 weeks, the mice sacrificed, and the organs were separated. During the EGCG and the 5-fluorouracil injection days, we could see that EGCG showed less impact on the weight of the mass compared with the 5-fluorosyl group, which decreased day by day as the purple color has shown. So we may get a conclusion that EGCG may be less toxicity to the mass than the chemotherapeutics. And then we measured both the volume and the weight of the tumor from the mass. It seemed that after treated with drugs, the EGCG and 5-fluorosyl, the tumor becomes smaller. And then we use the HE staining to see the interior structure of the tumor. In the control group, the cell grew well, and in the EGCG groups, especially in the E20, we could see from here, the nucleus had condensed and even dispersed into the cytoplasm. And in the 5-fluorosyl group, the nucleus even dissolved and the cell had lost its structure. So both the EGCG and the 5-fluorosyl could damage the inner structure of the tumor. And finally, we examined the expression level of TNF-alpha and VEGF. TNF-alpha is a member of tumor necrosis factor and has been verified to be helpful if you have treated with chemotherapeutics or the radiotherapeutics. And the VEGF is an autocrine factor from the vascular itself, and it could promote the proliferation of the vascular endothelial cells and could enhance the permeability of the vascular. So it plays important role in the tumor proliferation and metastasis. The blue dot reflects the nucleus of the cell, and the red dot reflected the expression of the antibody, the TNF-alpha and VEGF. So after treated with these drugs, the expression of TNF-alpha enhanced and the expression of VEGF decreased. EGCG has showed anti-cancer effect in vivo. So totally, what I have presented today is just a small part of the whole study. But from this study, we concluded that EGCG have showed anti-proliferation and pro-apoptosis effect in vitro, and its anti-cancer effect also being tested in vivo. All of these studies have been finished last year in China, in Zhejiang University. It's located here in Hangzhou and quite near to Shanghai, just 1 hour drive distance. And here is a picture of my university. It's beautiful, and I'm come from the Tea Science Institute for the graduate student, and we also have the Tea Science department. As the Zhejiang University had become the top 3 universities in China, and among all the comprehensive universities in China, only Zhejiang University have the Tea Science department and enroll both the undergraduate student and graduate student. So if you would like to know more about tea, since Hangzhou is also the hometown of the West Lake Dragon Well tea, it's the most famous green tea in China. So if you would like to know more about tea, welcome to China and welcome to Zhejiang University. Thank you.
And so for our last of the 4 student speakers, we have Crystal Ye, an MS student in the Department of Chemistry, who's here working under the direction of Professor Jackie Gervais-Haig. Crystal? Crystal's topic is studying the natural products of tea, what science can bring to the 21st century. All right, welcome.
Thank you, Catherine, for the introduction. And as she correctly pronounced my Chinese last name, my last name actually means leaf, so maybe it is a sign that I should be studying tea leaves. So what exactly is 21st century tea? Well, as a student, or as a graduate student here, I study natural products chemistry, and when I think of 21st century tea, I think of food as medicine. So, you know, taking care of ourselves by eating good food and using that to prevent diseases instead of getting the diseases and then, you know, taking medicine to prevent itself. So a little bit about myself. I got my bachelor's in science in pharmaceutical chemistry here at UC Davis, And I originally wanted to go to pharmacy school, so I've always had a passion for drugs and pharmaceuticals and how those compounds affect our bodies. In my in my junior year, I joined Jackie Gervais-Haig's lab, and I really fell in love with organic chemistry and doing research. So I decided to pursue my master's in science in pharmaceutical chemistry, and I'm currently a second-year student. Before this experience, and even to this day, I cannot keep succulents alive. So it's really, truly amazing that I'm holding this tea plant here with such beautiful roots, and they're still living and thriving for our project that we have. So I like to call myself a pharmaceutical chemist. In many of the talks that we learned about given in these last few days, we know that these are the different chemical compounds that in our cup of tea. So one of the most popular ones that you may think of are caffeine, and these are the derivatives of caffeine, theophylline, theobromine, that are also found in a cup of tea. We also have amino acids such as L-theanine, and then as we heard in our talks today, we have EGCG, catechin, and theaflavins. And these are just very general groups that are in a cup of tea, and people do believe that these contribute to the health benefits of drinking tea. But when you think about a cup of tea, you're thinking of all these antioxidants and energy from caffeine, but you're not really thinking of lipids in tea. So that's what my research is really focused in, studying the lipids in tea. These are all just different groups of lipids that are found in tea and have been identified to be in tea. But I just want to note that these are just the general groups. There's a lot of differences in these structures that make these compounds have different biological activities. But the one that I'm most interested in my research are called, these compounds here, called the steryl glycosides. And these compounds have a carbohydrate group attached to a lipid group. And they've been found in the literature to have a lot of different biological functions. Independently, these compounds that were studied, not in tea, were found to have all these different biological functions in humans. So they can decrease the risk of cardiovascular disease, they have beneficial immune modulating properties, they're anti-cancer, and these are obviously all good things that we may want to consider when we're drinking our cup of tea. So one of my goals as a natural products chemist is to find these phytoceuticals in tea plants. So as we may know, that the only part of the tea that we're really consuming is the fresh 1-tip-2 leaf that's harvested from the top of the tea plant. That makes our cup of tea. But what about the rest of the plant? Because a lot of the older leaves and more mature leaves, it doesn't make for a really good quality cup of tea. But as a chemist, I'm interested in isolating those compounds, the different seeds, leaves, branches, even roots, to look for these glycolipid compounds and think about making pills as dietary supplements, or even to help treat those diseases that I mentioned before. So my job as a natural products chemist is I go into the field and I collect all these different compounds. So here are some leaves. This is, if you're not familiar, what a tea seed would look like. So on the outside is this nice green pod, and on the inside, you can't really see here, is a dark brown seed coat. It's very hard. And when I open up the seed coat, it reveals the gametophyte, and inside that is like an embryo. And so when I do that, I take all these compounds, I separate it out if necessary, and then I ironically put it into a coffee grinder. And I use the coffee grinder to grind up my sample to make it really nice and homogeneous, so when I'm extracting out the compounds, I can get a really nice, even extraction. And so I use organic solvents, especially because I'm looking for organic compounds. So I pour different organic solvents in order to extract out the different chemicals that I'm interested in these material. And this is what it looks like. So in this round bottom flask, I have a lipid extract of leaves. So you can just see it's this thick green mass of oil, and there's some chlorophyll and things like that in there. But how do I really know what's inside this round bottom flask? So one of the really cheap and easy tools that we have in chemistry is called thin layer chromatography. And using this tool, I'm able to see, and with the use of standards, I'm able to see that if these compounds are present or not in a cup of tea. And these TLC plates, they base how they work is based off of polarity. So they separate out these compounds based on their different polarities. So in each of these TLC plates, let's start at looking this one on the far left. This is my standard right here, and then this row is my crude, and this is a co-spot. So here we can all see that this type of compound, the stereoglycoside, is present in my crude. But we don't know exactly what that chemical structure is because different similarities, like example, like an addition of a double bond or an extra carbon, they will still have a similar polarity, so they'll show up the same. So this is just a quick way for me to see, is this compound present in my sample or not? And in our preliminary studies, we've been able to identify this compound present all the way from the seed, fresh leaves, processed tea leaves, brewed leaves, to our cup of tea. So we're actually consuming a small amount of this compound. And so this won't tell me exactly what that structure is, but we have tools to analyze these compounds. One of the tools is called mass spec, and the other one is called nuclear magnetic resonance. So this thing, I actually have to climb up this really tall ladder to get to. And basically, the basic principles of these is mass spec allows me, with the help of standards, to match the mass of these compounds. So if I have a crude sample that I inject into the mass spectrometer, it will give me a molecular weight of the compound. And when I compare it to the standards that our lab has made available, I can see if I do have alpha sigma stereolith compound here in my cup of tea or not. Another tool that we use to characterize it, because mass will just tell me the weight, a lot of compounds can have the same weight, but to really be able to identify that this is the correct structure, I use nuclear magnetic resonance. And that's essentially like an MRI instrument where they utilize radio frequencies, and the different carbons and different atoms on this structure resonate at different energy levels, and this allows it to produce a spectra where I can look at and characterize and figure out that, okay, this is the exact compound that I have. So putting all those tools together, I'm able to really understand and better understand what is exactly in those lipids in our tea and see what those phytoceuticals actually are. So this is a magazine cover of CNN beginning of this month, and it says here, food as medicine, ? And with my project, I would like to be able to identify and tell you exactly what is in that crude, but I have to purify out those compounds. But in the future, we really want to be able to understand, here's your cup of tea. These are the compounds that are present in it, and these are how they will help you be really healthy. So to summarize, natural products are an underused resource for pharmaceuticals and drug discovery. Science can help us better understand what truly makes a cup of tea healthy. And that's all I have for today. So thank you all for your attention.
I will just say, see why we love our students. See how inspiring they are. See how impressive they are. At their various level in their various departments and disciplines, and each with a wonderful research question that is the driving force behind this university and behind GTI. So let's give them all a round of applause. Okie dokie. So if there are questions, please step up to the mics. I'll facilitate. You took our breath away, you guys. Here comes a question. Please speak into the mic, though. All right, that works.
In that last talk, talking about lipids in tea, I'm kind of curious because in at least the tests that we've done, lipids in tea, from my understanding, are it's a very, very small percentage of what's in the leaf. And especially if you're drinking a cup of tea, typically when you consume fats, it's on the order of grams. And whenever you drink tea, you're using only a few grams of tea leaf to begin with. So if you have a very small percentage of that being lipids, I'm just wondering how effective those can be. Or I guess my question could also be are you taking the lipids from the tea leaf and just concentrating it and like gathering a whole bunch from a whole bunch of tea leaf? And then if so, is that a sustainable source for producing pharmaceutical products?
Thank you for your question. So I guess to address that last part of the question, when we do our natural product studies, you're right that these lipids are in very small quantities. So when we do extract them, we have to take material in a kilogram scale. But as for the pharmaceutical aspect, plant phytosterols are very beneficial to help lower like cholesterol absorption. So the recommended dose for like an adult is 2 grams a day. So if we were to take the unused sources of the tea plant to make them into, pharmaceuticals and pills and things like that, I think it's still going to be very viable in that way, because those parts, like we know that some farmers, routinely prune their plants, and when they do that, a lot of that goes into compost or just waste. So if we it's just thinking about utilizing every part of the plant to make something useful for it. And then as for like what actually you're consuming in a cup of tea, we don't really know that yet. That's still being studied, but really good question.
Next, over on the Raj.
Hey, is this working? I don't have Noli's voice.
Yeah, we can
so
hear you just fine. Thanks.
My question is also for Crystal. I was curious, my understanding from your presentation was that mass spec is a way that...
Speak into the mic.
Okay, sorry. That mass spec is a tool that can be used to track compound presence of all compound types. And if that's the case, if UC Davis was entertaining the possibility of providing that to industry people as well.
What is your question exactly?
Yeah, sure. Is mass spec a service that could be provided to other outside organizations from the UC Davis system?
Oh, yeah. So we do have mass spec facilities here on campus, and they do collaborate with, outside industry members, Davis members, like private business owners also. So right now on campus, we have the the mass spec core facility. There's a website for that, and then there's also the West Coast Metabolomics Center at the Genome Center, and they have fabulous mass spectrometers to analyze samples.
Thank you.
More questions? Any questions? Well, then in that case, let's thank our speakers one more time.